Epinephrine is an example of GPCR communication. Epinephrine is a ligand that binds with the G protein-coupled receptor. This causes the G protein to bind with the receptor. The GDP in the G protein is replaced with GTP. Then, the activated G protein diffuses across the membrane and attaches to adenyl cyclase enzyme. This enzyme is now activated as well. Adenyl cyclase then converts ATP to cyclic AMP. CAMP is a secondary messenger that further activates other secondary messengers through phosphorylation. The final activated messenger activates enzymes that carry out cell response. Cyclic AMP produced from epinephrine synthesizes the enzyme of glycogen phosphorylase. This enzyme is responsible for the cell response of breaking down glycogen into glucose monomers that power our flight or fight response.
Capsaicin activating TRPV1 signaling pathway is an example of an ion channel. Capsaicin enters the cell by simple diffusion and then binds to one of the transmembrane domains of TRPV1, which opens the ion channel. The opening of the TRPV1 channel allows calcium ions to enter the cell, leading to the activation of the enzyme calcineurin. Calcineurin removes phosphate groups in the phosphorylated form of the transcription factor NFAT2, which is typically found in the cytoplasm. Once dephosphorylated, NFAT2 moves into the nucleus where it blocks the transcription of another protein, ATF3. ATF3, when active, prevents the cell cycle regulatory protein, PGOG, from inducing apoptosis and blocking cell division.
In this example of RTK, an epidermal growth factor binds with an epidermal growth factor receptor embedded in the plasma membrane. This binding causes the inactive receptor tyrosine proteins to come together. This becomes partially active. Then, a phosphate from 6 ATP molecules are transferred to each amino acid tyrosine on the EGF receptor. This phosphorylation of the receptor fully activates it. Active relay proteins, in this case MAPK kinase, go through a phosphorylation cascade and ultimately are used to transcribe genes for cell growth, phosphorylate other transcription factors, or phosphorylate other proteins found in the cytosol.
Aldosterone is an example of intracellular signaling. Aldosterone diffuses through the capillary membrane because it is hydrophobic. Aldosterone binds to the mineralocorticoid receptor in the cytoplasm, activating it. The hormone receptor complex then diffuses into the nucleus and binds to specific genes. The bound protein acts as a transcription factor, stimulating the transcription of the gene into mRNA. The mRNA is translated then into a specific protein.